Chapter 7, Cell Reproduction, Part 4. We've been looking at meiosis in a rather generic way, a kind of a basic model for how meiosis works. But now we can look at the individual styles of making gametes in both males and then females. And they're quite different. At the end of meiosis, 4 haploid cells have been produced, but the cells are not yet gametes. They need to become mature gametes capable of fertilization. The development of diploid cells into gametes is called gametogenesis. It differs between males and females. A gamete produced by a male is called a spermatozoon, and the process that produces a mature spermatozoon is called spermatogenesis. I'm trying to use the term spermatozoon as often as I can to specifically refer to the mature gamete, and it's because there's so many terms of the intermediate referring to the intermediate cells. I want to not have it be more confusing. So a gamete produced by a female is called an ovum. You know this as an egg, and the process that produces a mature egg is called oogenesis. Spermatogenesis occurs in the wall of the seminiferous tubules of testes. Immediately inside the tubules are diploid undifferentiated stem cells called spermatogonia, or singular spermatogonium. Spermatogonia are germ cells. So they're stem cells, meaning that they can be convinced or teased into becoming all different sorts of cell types. They're not committed to being a certain cell type, and they're germ cells because these are the special kind of cell that is either going to give rise to gametes or is a gamete. The germ cells are different than the other type of in the major category of body cells or somatic cells, which are everything else like your heart, your eye, your skin, for example. The stem cells that form spermatozoa exist during gestation and are present in an inactive state at birth through the beginning of adolescence. They undergo mitosis, and of the pair, one of these spermatogonia cells, now called the primary spermatocyte, goes on to differentiate into spermatozoa and emerge into the lumen of the tube of the seminiferous tubule. And let me see if I can jump ahead a little bit. I'll jump a little ahead here and kind of give you an idea of where we're heading. Spermatogonium is going to duplicate, and then one of the cells goes through meiosis 1. So we'll go back over here again, and we'll say the primary spermatocyte then goes through the first meiotic division, and a haploid cell is produced called a secondary spermatocyte. But the haploid cell must go through another meiotic division. So you know this from your basic knowledge of how meiosis works. We have cells that are haploid, but they have extra genetic material. In other words, the chromosomes have 2 arms and 2 legs, if you can picture that, like I've said before. But the arms and legs are carbon copies of each other. So we can have one chromosome that has 2 arms and 2 legs, or we could pop it apart at the centromere, and now we can have 2 chromosomes composed of 1 arm and 1 leg, but they carry the same genetic material as the double 2 arms and 2 legs chromosome. We're just taking the same information and distributing it into 2 different cells ultimately, but it's the same information. So the diploid cell has duplicates of the information, and the haploid cell has 1 copy of the information, which is plenty to go on to become a gamete. Okay, so at adolescence then, gonadotropic hormones from the anterior pituitary gland activate these cells, resulting in the production of viable spermatozoa. So there are a lot of these cells that are waiting to be triggered by hormones, and then the spermatozoa are produced then at that time. The cell produced at the end of meiosis is called a spermatid. But when it reaches the lumen of the tubule and grows a flagellum, and also some other things like special enzymes on the cell end that will help in penetrating an egg, it is called a spermatozoon. So we have 4 of these spermatozoa that result from each primary spermatocyte. And here's the picture here. So spermatid isn't quite ready yet because it needs to become modified, mature. They call it differentiation, but mature into this spermatozoa that has a tail. Notice how little cytoplasm there is here. It's hard to have a comparison right now, but these cells have very little cytoplasm. Really, they're DNA delivery mechanisms. So here's a way that you can look at this. Here's a cross-section. I think this is maybe a rabbit testis here. And this is a graphic. It's in a different language. I don't know. It looks like it's Greek or something like that. Oh, no, it looks like this is in English. Okay. But you can see that you can compare the two. If you look at the figure on the left, you can watch those large cells, perhaps picturing that they're dividing, moving toward the lumen, getting their tails, and then they'll exit through that lumen. Oogenesis is something that produces gametes in the outermost layers of the ovaries. It starts with a stem cell, which is a germ cell, because it's going to make gametes. It's called an oogonium, or plural is oogonia. The cell undergoes mitosis, eventually resulting in up to 1 to 2 million oogonia in the embryo. So this is different. We really have many more of these cells that are waiting in the ovaries of the embryo, the female embryo. And now we're going to have one of these cells that's going to start through meiosis. This is called a primary oocyte. But at the time of birth of this little girl, all future eggs are arrested in the prophase 1 stage. So they're there. They've been made and stored in the ovary. And then at the time of adolescence, the anterior pituitary gland hormones cause the development of a number of follicles in an ovary. This results in the primary oocyte finishing the first meiotic division. So let's see what that looks like here. Let's see the picture here. Okay, so we have an oogonium that undergoes meiosis, and then we have lots of those, maybe millions of those in the ovary, and they're hanging out in prophase 1. Okay, now, after puberty then, the meiosis continues. It's triggered to continue, and we'll see what happens next. Okay, so we go back here and we see that we have the primary oocyte the cell of interest here. And this cell divides unequally, with most of the cellular material and organelles going into one cell, now called the secondary oocyte. This has one set of chromosomes now. Now we're going from a diploid cell to a haploid cell, and a small amount of cytoplasm goes into the other cell. So we have one big cell. I don't really say it very well here. We have one large cell and one small cell called the polar body. Now think about this. If we had two cells that were dividing from diploid to haploid, then that's fine, because either one of those haploid cells could join with a spermatozoon, and then we could have the first cell of a new life, right? But if we're only keeping half of the ones that are being produced, that is that's a spot of like throwing the dice. Which one of the cells is going to have the potential of being fertilized? And right away, we just have one of the cells that dies, but which one dies is, as far as we know, random. And I wouldn't be able to tell you why that happens, but it does happen. Now we have, after this secondary oocyte is formed, then we have another pause, And this pause occurs at metaphase 2. And at metaphase 2, then we have this kind of a tension on those chromosomes. They're being held in suspended animation, let's say. They're not going through the phase, but they're being arranged in the cell with the cytoskeleton holding them there at the plate, the metaphase plate in the middle. So at ovulation, the second oocyte will be released from the pause and travel toward the uterus through the oviduct. So it didn't say it here, but we know that ovulation is going to occur at puberty. And then the secondary oocyte has the potential of being fertilized in the oviduct. If it's fertilized, then it continues through meiosis too. So it's fertilized before the whole meiotic system is finished. Now, there's another spot here where we have a second polar body produced. So what's happening is the cytoplasm is being concentrated into one large ovum instead of having evenly balanced daughter cells being produced. Okay, so then at the end of this meiosis 2, which is ongoing as the egg is traveling, we have a fertilized egg, fertilized ovum, that now has all 46 chromosomes. We have 23 from the father and 23 from the mother, and we now would be on our way to producing, we have the first cell of a new life, basically, and this is what we would call the zygote. And here's oogenesis. Again, the oogonium, we have mitosis, and we have this kind of a suspended animation in prophase 1. Meiosis continues. The primary oocyte divides into a secondary oocyte and a polar body. And ovulation then, and perhaps sperm being present in the fallopian tube, and then it continues, the secondary oocyte continues to develop, and then another division occurs, and another polar body exists, and then we end up with one fertilized egg. And that egg, it doesn't show in this picture, but the egg would be very large. We can kind of see the difference between the sperm and the egg, but it's not realistic. The egg is much bigger than the sperm, for sure. And then these numbers here, 2n and 1n, the 2 represents a diploid cell, and the 1 represents a haploid cell. And here's something a little bit more fun to look at for oogenesis. We have a little graphic here that shows you can kind of picture the egg. You're not seeing all the duplication here, but you can kind of picture the process of the egg, going through its phases and finally maturing enough to be released from the ovary into the fallopian tube. So this is the stage at which the egg would be ready to be fertilized and continue meiosis or oogenesis. And then here's a microscopic slide showing the follicle that is containing the egg that will be ejected from the ovary, and it probably come in this direction. You can see there's an opening here. It goes into making it to the fallopian tube. Mitosis versus meiosis. Both mitosis and meiosis result in eukaryotic cell division. The primary difference is the differing goals of each process. The goal of mitosis is to produce 2 daughter cells that are genetically identical to the parent cell. It happens when you grow or repair. So this is what mitosis is, the process by which you would heal a wound, replacing cells with the same kind of cell. As you're growing, where most of us aren't growing anymore, we're kind of maintaining what we have, but mitosis would replace cells that are aged or damaged. But in an individual that's growing, that's how people get taller and bones lengthen, etc. The goal of meiosis is to produce sperm or eggs, also known as gametes. The resulting gametes are not genetically identical to the parent cell. Gametes are haploid cells with only half the DNA present in the diploid parent cell. This is necessary so that when a sperm and an egg combine at fertilization, the resulting zygote has the correct amount of DNA. Okay, so here's a little graphic that you can look at if you want from your textbook. Chromosomes disorders, changes in chromosome number. This can happen during these oogenesis or spermatogenesis, resulting in sperm or egg that are not balanced in their number of chromosomes. What would happen if an entire chromosome were missing or duplicated? What if a human had only 45 chromosomes or 47? This is a real possibility, usually due to mistakes during meiosis when chromosomes do not fully separate from each other when sperm or eggs are being formed. Specifically, non-disjunction occurs when homologous chromosomes or sister chromosomes fail to separate during meiosis, resulting in an abnormal chromosome number. Non-disjunction may occur during meiosis 1 or meiosis 2. Most human atypical chromosome numbers result in the death of the developing embryo, often before a woman even realizes she's pregnant. Occasionally, a zygote with an extra chromosome can become a viable embryo and develop. Trisomy is a state where humans have an extra autosome. Autosome is a chromosome that is any one except for a member of the pair of sex chromosomes. So autosomes are the pairs 1 through 22, and then the 23 pair of chromosomes, either XX or XY, are called the sex chromosomes. So trisomy is a state where a human has 3 of a particular chromosome instead of 2. For example, trisomy 18 results from an extra chromosome 18, resulting in 47 total chromosomes. To identify the chromosome number, a sample of cells is removed from an individual or a developing fetus. And then metaphase chromosomes are photographed. So metaphase chromosomes are thick and easier to see than other chromosomes. So these are used to produce something called a karyotype, which displays any abnormalities in chromosome number. So you can actually see the chromosomes and count them and see how many there are. And sometimes there can be abnormalities, like chromosomes that have broken off have pieces broken off, for example. Trisomy 16 is the most common trisomy in humans, occurring in more than 1% of pregnancies. This condition, however, usually results in spontaneous miscarriage. The most common trisomy in viable births is trisomy 21. I'm also going to remind you that at the end of the semester you'll have a poster project. So some of these topics might be of interest to you to dig into a little bit more with a poster. This figure here shows you how non-disjunction can work. And we start with our primary cells here that are going to divide. And we can see that in the bottom one, we have the cells that are pretty even. They're evenly distributed. And as we go through the second division here, what we're seeing is that one of the chromosomes doesn't split apart at the centromere like it needs to. And so what we end up with is a cell at the end here that has actually has one normal chromosome and one duplicated chromosome. So the duplicated information in a chromosome can be lethal. And at the bottom here, we have something that turns out to be fine, because we have the chromosomes now that become evenly distributed in the gamete. And on the top, we have another situation where we have more of the chromosome number going into the top cell here. And then when they divide, it divides again in meiosis 2. You can see that the gametes have too many chromosomes. Each gamete should have basically 2 according to this schematic here, we'd want to be a normal 2-chromosome gamete like what we see here. But in the top, we see some of the gametes have 3 chromosomes and some have 1. So if we took a chromosome like this on the top and we fused it with a gamete that had 2, then we're going to have too many chromosomes in that zygote. And that's going to be that'd be a problem. It can't be, the chromosomes cannot speak to each other. So in a kind of an analogy, they need to be able to interact properly and line up properly. If there are too many, then that causes problems.